Okay, welcome to part 2. We're going to be starting central dogma. So what is that? It's the process of transcription and translation to go from DNA to RNA to proteins. So in this picture to the right, we've got our replication, which we just went over in the last video. And then we're going to go into transcription, which is taking DNA and making it into RNA. So once we have our RNA, we can do translation, taking that RNA and making it into proteins. So there are 3 types of RNA which are all made in the nucleus. The first one is messenger RNA or mRNA. It's a single and straight strand that transmits DNA information and serves as a template for making the proteins. So here is that's our mRNA. Our transfer RNA is tRNA. It's a single folded strand. It's got a complementary base pair to this codon here, and it's involved in protein synthesis, bringing amino acids. And then our last one is ribosomal RNA or rRNA. It's a globular form and it's part of the ribosome structure. So this rRNA will help make up the ribosome. This one. Okay, so transcription, making mRNA. So the mRNA molecules are made by copying part of the DNA. The process of DNA to RNA again is called transcription. So mRNA is really being transformed. So where there's normally a T, you're now going to replace it with a U. So if we take a look at that, what does that actually mean? If we break down our base pairing, normally when you're looking at DNA to DNA, your A's pair with your T's, your G's with your C's. If you've got a T and A will go there, and then your C's and G's. But in transcription, you're going from DNA to RNA. And so your A will actually bond to a U. So we're replacing that T with a U. Your G's and C's are normal. If you have a T in DNA, the RNA will still be an A, and then C to G. So again, this is DNA and this is RNA. So in RNA, our T's are not there, they're U's. Okay, so if we look at an overview of transcription, it's happening in the nucleus. Oh, rats. Here's our nucleus, right? And the RNA copy of a gene is made there, and then it can move out of the nucleus into the cytoplasm. So once it's in the cytoplasm, then it can be used to start making proteins. And just a little bit more information for you. The enzyme that makes the strands of DNA is DNA polymerase, and the enzyme that makes the strand of RNA is RNA polymerase. So when we did DNA replication, we talked about DNA polymerase, making strands of DNA, and now RNA polymerase will be making those RNA strands. So when RNA is getting made into the mRNA, it'll be edited sometimes, so there might need to be some editing before a final draft can go through. So we have introns, which are removed. So think about your introns as intruders. You want to get rid of your intruders, right? And exons stay. So think about exons being excellent. You want to keep the things around that are good. Exons stay, they're excellent. So in this DNA strand, we have our exon, which stays, and our intron, which will leave. So when our RNA gets made from our DNA, we still have these exons and introns, but when we edit it to make mRNA, we keep just the exons. Notice that our introns are not there. It's just the exons. Okay, so let's try our hand then at transcription. So here we've got our DNA, right? And if you remember, when you have a T in DNA, you have to replace it with a U in RNA. And so we are going to make RNA based off of this DNA template. So our T should become a U, right? Oh, wrong, A. Our A will become a U, and our C will become a G. And then this will be a G, this will be a G. Our A will become a U, C, U, U, U. Now this will be an A, G, C, U. Nice. That's funny. I literally was like, your T will become a U. So if your T will bond to an A, but your A would normally bond to a T, right? If it were DNA, but those T's have to become U's. Okay, we got this. So RNA has letters and words. So the letters of mRNA are A, U, G, C. Just went over that. And the words of mRNA are called a codon. And each codon is 3 letters long, and each codon codes for an amino acid. So here we've got U, U, C, which is our codon, and it codes for phenylalanine. Cool. And there are special codons, so there are there's a start codon, which is AUG, which is methionine, and it's the first amino acid of the protein. UAA, UAG, and UGA are all stop codons, so they will actually act like a period at the end of a sentence. The stop codons will stop protein production, so it's done. And if we look at this picture to our right, show you how to use this here. If you're looking at the beginning of your codon, let's say it starts with A, here's our A, and the next letter is U. We go over to the U, and then the last one is G. So then, boop, we go to G, and there is methionine, our start codon. If we were to look at let's say UGA, we find the big U, so that's here. Then we go to the G, which is there, and then that last letter is A, so we go here, and that is stop. So that's how you use an amino acid chart, and yeah, that's about it. Okay, so proteins, why are they so important? Why do we have to talk about translation, transcription and translation? Well, everything in your body is made up of proteins or made by a protein. So if you remember keratin, that adds strength and stuff to your hair and your nails. And hemoglobin is a protein in your blood that helps transport oxygen. And then this last one, insulin. So remember that proteins are made of amino acids linked together in a polypeptide chain. So here we would have our amino acids linked together in our polypeptide chain. And so to get to proteins, we look at our genetic code. So remember, there are 4 letters in RNA's alphabet, the A, C, G, U. But there are 20 letters in a protein's polypeptide alphabet. So how do we get from the letters to a polypeptide chain? Well, we have to use translation. And so next, we go through the assembly of proteins at the ribosome. So here's this sweet little comic for you. All right, hopefully you had time to read that. So moving on to translation then. During translation, the cell uses information from the mRNA to produce the proteins. So on the ribosome here, the tRNA, this guy, will bind to the appropriate spot on the mRNA. So it'll bind right there, that's its spot. And then the next tRNA molecule will bind to the next codon on the mRNA. And the ribosome then will link the two amino acids. And after each tRNA has dropped off the amino acid, it will disconnect, boop, and the process will just keep going. So more tRNA will come and land, the ribosome will move along and pump out the amino acids, and then this one will leave, and it'll just keep on going until the stop codon is reached. So more about tRNA. So this one, its job is to know which codon goes to which amino acid. So each tRNA molecule has an amino acid attached to one end. So 3 unpaired bases at the other end, the anticodon. So here's the codon on the mRNA, and then the tRNA is carrying the anticodon. And these are the bases that are complementary to this mRNA codon. Okay, so on the ribosome then, the tRNA will begin to bind in its appropriate site, right? We already said that. And then the next tRNA molecule binds to the next codon on the mRNA. The ribosome links those again. And after each tRNA has dropped off the amino acid, it disconnects and the process just repeats. So we kind of already said that up here, but there's another picture for you. Okay, so with the growth of a polypeptide chain, the polypeptide chain will just keep growing until a stop codon is reached. So remember that's a stop codon is UAG, UGA, or UAA. So when it reaches a stop codon, then the polypeptide chain gets released so that we end up with this little setup right here. So here we have our start codon, AUG, and then we've got these other codons coding for these amino acids, and then here's our stop codon, so we no longer are producing the polypeptide chain. That's the end of it. So if we look at this really big picture that kind of overviews what we just walked through, we've got our DNA over here, and we're going to go through and do the process of transcription. So we make this messenger RNA copy so it can leave the nucleus. This whole region here is the nucleus. Right, so the messenger RNA leaves the nucleus, and then it gets used by the ribosome here, and in translation, the tRNA then over here can come and take those anticodons and bind to the appropriate spot on the mRNA, and then when the ribosome comes, it joins these amino acids to form this long polypeptide chain. So translation also takes place in prokaryotes, but it's a little different. So polyribosomes are a single mRNA that can be translated by several ribosomes at the same time. So in this picture here, we've got our DNA strand here and all these RNA polymerases on it that have these polyribosomes. So we've got all of these polyribosomes that are working to translate instead of just having one that's running down the entire length of the DNA or the mRNA, excuse me. We've got multiple ribosomes. Okay, so what happens then when we have all of these polypeptide chains? After translation, the chains of the amino acids will fold into very specific 3D shapes to perform their biological functions, and these shapes will depend on the interactions among amino acids via hydrogen bonds. So if we take a look here at the primary structure, at this one, we've got our polypeptide chain of these amino acids, and then for the secondary structure, there are these hydrogen bonds that are going to cause it to either go into the alpha helix, or down here we've got this pleated beta sheet, or beta sheet here. So there are these hydrogen bonds that are bonding with oxygen there, and this one with that R group. And then we'll have our tertiary structure. So here is our beta polypeptide. And then those will come together to form the quaternary structure. And so here we have the protein hemoglobin that's going to carry our oxygen through our blood. Okay, so that's the rundown of the central dogma. We went from transcription, doing DNA to RNA, DNA to RNA, and then we looked at translation, going from RNA to a protein. All right, if you've got any questions, let your teacher know and they'd be happy to help you. Alrighty, thank you.